manufacture food for their development and growth. In addition, they also provide food for us, making available the nutrients essential to our existence. The animals that supply us with meat also derive their food from plants. We know that it is through the process of photosynthesis that green plants produce the food so vital to us. Let's review briefly the fundamentals of photosynthesis. A plant such as corn absorbs water, H2O, from the soil. It also absorbs dissolved minerals from the soil. The plant, in addition, takes in from the atmosphere carbon dioxide, CO2. Using the energy of sunlight and using chlorophyll, the countless molecules of green pigment in leaves that give them their color, the leaves manufacture certain chemical products. The carbohydrate called glucose, a simple sugar, is one of the most important. Some amino acids, protein materials, are also manufactured. Photosynthesis produces, as a byproduct, oxygen, which is released into the air. Now let's consider the carbohydrate product, glucose. Can we prove that green plants produce this simple sugar? From this green plant, we have taken some bits of leaves. The finely cut pieces are in the test tube. One test for glucose and other simple sugars uses this blue solution called Benedict's reagent. We add Benedict's reagent to the leaves. Then we heat the tube. After a few moments, the blue solution turns a reddish color. This test indicates that a simple sugar is present. Evidently, glucose has been produced in the leaves. Here's a test to determine if oxygen is given off during photosynthesis. A glass funnel and a test tube are arranged so that any gas given off by the green plants in the funnel will be collected in the tube. Gas, indicated by these bubbles, is escaping from the plants into the tube. After several hours, we cork the test tube and remove it from the beaker. Now we'll use a standard test for oxygen, a glowing splint, to see if the tube contains this gas. The flame indicates the presence of oxygen. Now, let's examine the structure of a leaf and see the part it plays in the process of photosynthesis. In the blade of the leaf, the veins transport water, dissolved minerals, and foods between the green cells of the leaf and the stem. From the underside of the leaf, we'll strip off a thin layer of leaf tissue, the epidermis, and prepare a slide. Under the microscope, the openings called stomata and their associated guard cells may be seen. It is through these openings, the stomata, that the carbon dioxide used by the leaf enters and the oxygen produced by the leaf diffuses out. Here, a portion of a leaf has been placed between two sections of pith. A cross-section of the leaf can be prepared by slicing through the pith and the leaf. The leaf section is then viewed under a microscope. Both the upper epidermis and the lower epidermis generally consist of single layers of cells. Between the upper and lower epidermis are the mesophyll cells, which include this layer of palisade cells and these spongy cells. Within the mesophyll cells, we find chloroplasts, and it is in the chloroplasts that photosynthesis occurs. Here we see the green chloroplasts moving within a living leaf. Let's see what happens in the chloroplasts during photosynthesis. You remember that one of the most important products of photosynthesis is glucose. In order for glucose to be produced, First, light energy must be changed to chemical energy. The light energy is absorbed by the chlorophyll molecules within the chloroplasts. Chlorophyll molecules, represented by these ovals, are large molecules. Several kinds of chlorophyll are known. A molecule of chlorophyll A consists of 55 atoms of carbon, 72 atoms of hydrogen, 5 atoms of oxygen, 4 atoms of nitrogen, and 1 atom of magnesium. Also complex is chlorophyll B. The formula is slightly different. Chlorophyll molecules become activated as they absorb light energy. The energy of the activated chlorophyll molecules is used to rearrange the atoms which make up the carbon dioxide and water molecules. Here is one interpretation of the way it happens. These represent 2 water molecules. Each consists of 2 atoms of hydrogen and 1 atom of oxygen. The light-energized chlorophyll splits the water molecules. 2 atoms of oxygen unite to form 1 molecule of oxygen gas, which is released to the air. In addition, molecules of carbon dioxide, CO2, are split. 1 oxygen atom from a molecule of carbon dioxide unites with 2 atoms of hydrogen from the water molecules to form a new molecule of water. The remaining atoms of the carbon dioxide unite with the remaining hydrogen atoms to form CH2O, a basic unit of the glucose molecule. The formation of the glucose unit is perhaps the most important result of photosynthesis. Until we have further evidence as to the exact manner in which the glucose unit is formed, this chemical equation represents the overall reaction. However, since water is consumed during the reaction, the net result is this. The CH2O is a unit of the glucose molecule. 6 CH2O units join together to form C6H12O6, the glucose molecule. Using it as a basis, the plant is able to manufacture all of its other constituents. This net equation summarizes what the plant accomplishes during photosynthesis. From the carbohydrate glucose, plants are able to manufacture additional carbohydrates, as well as proteins, fats, and vitamins. We have just dealt with the basic steps in photosynthesis. The process actually has many intermediate steps. More than 30 steps in the overall reaction are known. One way plant physiologists seek to learn more about the production of glucose and the other products of photosynthesis is by the use of radioactive tracers. During successive stages of photosynthesis, plants are fed with carbon dioxide containing radioactive carbon. Then finely cut leaf samples are prepared and analyzed. Study of the samples shows that the leaves in the photosynthetic process are capable of absorbing and retaining radioactive carbon supplied in carbon dioxide. In a much simpler experiment, these students are studying another aspect of photosynthesis, the effect of light intensity. They are working with artificial light. It, like sunlight, will bring about photosynthesis. A light meter is used to measure the intensity. Under the influence of the light, the plants in the inverted funnel are producing bubbles which we know contain oxygen, a byproduct of photosynthesis. Now we'll increase the intensity of light as the meter indicates. With more light, there is an increased rate of photosynthesis as indicated by the increased bubble production. Where more accurate measurements of the rate of photosynthesis are required, the bubble count method is replaced by more refined procedures in a photosynthesis laboratory. This measuring instrument, a manometer apparatus, indicates rate of oxygen production. Changes in the levels of fluid in the tubes can be measured to determine exact amounts of oxygen produced. Returning to the bubble count method, we'll test the effect of different colors or wavelengths of light. Using a red filter, we find that red light gives rapid oxygen production. When we switch from red light to green light, keeping the light intensity the same, we see that the wavelengths of green light are much less effective in photosynthesis. Knowing which wavelengths of light are most effective in photosynthesis, plant physiologists also study how these wavelengths are absorbed by different pigments in the plant. A graph called an absorption spectrum reveals which pigments absorb certain wavelengths. Analysis of a chlorophyll solution confirms that it is the chlorophyll molecules that most effectively absorb and utilize light energy. We know that the process of photosynthesis involves the changing of light energy within the leaf to chemical energy. The basic energy transferring chemical compound used by a plant or any other living organism in the processes of food making, growth, and other life functions is ATP, adenosine triphosphate. It is believed that within the chloroplasts of the leaf, ATP is synthesized from two other chemical compounds, ADP, adenosine diphosphate, and P, a symbol for inorganic phosphate. We can represent the reaction this way. ADP and P, making use of light energy, form ATP. When ATP is broken down, ADP and P are formed again, and chemical energy is released. It is this chemical energy which is the source of energy for the rearrangement of CO2 and H2O to form the glucose unit CH2O and oxygen, which is released. From this glucose unit, the plant manufactures the foods that are stored in its various parts. It is upon such plant foods that we depend for our existence. So, within the plant, the process that produces the basic food compounds upon which almost all plant and animal life ultimately depends is photosynthesis.